a story about how to improve plant photosynthesis. Okay, please, Dr. Liu.
Hello, can you hear me?
Yeah, I can hear you, yeah.
Okay, great. Hello, everyone. I'm Looney Liu from University of Liverpool, United Kingdom. It's my great pleasure to be nominated by colleagues and be selected by the committee to give a talk about our research in my research group. So I have long research interest in molecular mechanisms of the photosynthesis and photo adaptation in different photoautotrophs using multidisciplinary techniques. For example, molecular biology, biochemistry, synthetic biology, structural biology, and computation modeling, for example. So my research group study the structure and functions of individual photosynthetic protein complexes from purple photosynthetic bacterial, cyanobacterial, algae, and plants to explore the evolutionary relationships and the physiological adaptation of photosynthetic systems. At native member level, we study also the architecture of native photosynthetic membranes and how protein building blocks are functionally organized and interact with each other in the native membranes to conduct efficient photosynthetic electron transport using different microscopy, for example, atomic force microscopy and electron microscopy. And we also examine the subcellular organization and biogenesis process of photosynthetic organelles and photosynthetic membranes into the cells of the photosynthetic organisms. So my research vision is to advance our current knowledge and understanding of a photosynthetic system developed in nature for efficient energy transduction and carbon fixation, and then inform the design and engineering of photosynthetic membranes and organelles or pathways to enhance plant photosynthesis. So we all know photosynthesis is essential for all life on Earth and also the sustainable growth of plants. But in nature, photosynthesis is not highly efficient. A major rate limiting step is the carbon fixation catalyzed by the enzyme called rubisco, which is the most abundant protein on Earth and also the major proteins in the plant leaves. So rubisco is not an efficient enzyme because first, it has a low carboxylation rate. And second, rubisco can interact with CO2 and oxygen. So by binding with CO2, it characterizes the carboxylation process to produce sugar. But in the at a high level of oxygen, they can also bind to oxygen to carry out the oxygenation process to have a wasteful process called photorespiration. So as I mentioned, at high oxygen levels, so rubisco has a very low specificity for all CO2 than oxygen. And because of the poor capacity to recognize the two molecules, most of the C3 plant rubisco has a higher error rate to distinguish CO2 and oxygen. So researchers try to test improving the CO2 level around the plant to improve the plant growth, and it works. But in fact, in practice, we cannot provide high CO2 in the field to improve the crop yield because of the climate change and the greenhouse effects, right? So an innovative strategy is to increase the CO2 in the cellular level, for example, in the chloroplast where the rubisco occurs and functions. So in fact, nature has already evolved various CO2 concentrating mechanisms in different organisms. For example, the carboxysomes in cyanobacterial and the proteobacteria, and pyrenoid in algae, as well as C4 pathway and CAM pathway in many plant species. In contrast, most of the crop plants don't have efficient CCM systems because they use the C3 pathway. So my group is of interest in the carboxysome-based CO2 concentrating mechanisms we call CCM. The carboxysome is a nanoscale protein-based organelles, and it structurally is resembling the virus capsids. As you can see, they have multiple copies in the bacterial cells. And carboxysome are the central CO2 fixing organelles inside the bacterial CCM, together with bicarbonate transporters in the plasma membrane and NDH1 complexes in the in the thylakoid membranes for bicarbonate uptake. So carboxysome encapsulates rubisco and carbonic hydrates within a polyhedral protein shells. So the shell is permeable to bicarbonate. So the bicarbonate can passively cross the shells to be accumulated inside, where in the carboxysomes, the bicarbonate can be converted by the carbonic hydrates into CO2. So in the local environment, there's a high concentration of CO2 around rubiscos for improving the rubisco activities. So that's a growing interest in engineering carboxysome-based CCM systems into the chloroplast to enhance photosynthesis. And also, we know that in cyanobacteria, the CCM has been very powerful at the global scales to contribute significantly more than 25% of the global carbon fixations in the marine and the freshwater ecosystems. So it's a very promising target for engineering crop plant photosynthesis. But since the discovery of the carboxysomes 17 years ago, there's still a big knowledge gap in the carboxysome research and also engineering carboxysome in other organisms. For example, the major question is how different building blocks assemble together to form functional entities. So my research group has recently conducted extensive studies on the carboxysome based on mainly focusing on stoichiometry compositions and the rubisco organizations and shell organizations and encapsulation mechanisms and the shell permeability functions and also engineering of the carboxysome in plant and ecosystems. So we use a model system called Helicobacillus nipolitanus, which is a chemoautotrophic bacteria. So the carboxysome in this Helicobacillus, the genes are mainly in a single CSO-S2 genomic operons. Along the operon, there are other bicarbonate transporters or activists or assembly factors that are next to these operons. So our first interest is the protein compositions. So we developed a method to purify high quality of carboxysomes and analyze using the biochemical methods together with Q-concat-based absolute quantification based on the mass spec analysis. So we can identify the exact copy numbers of the carboxysome proteins in one carboxysome. For example, there's a shell protein that has more than 1,000 copies, and rubiscos, there will be more than 400 rubiscos. It can be encapsulated in this nanoscale nanoreactors. So the information is quite important for fundamental understanding of the assembly, but also engineer the functional carboxysomes with proper protein abundance. So based on the purification, we also try to understand the overall 3D structures and the rubisco organizations of the carboxysomes using cryo-electron tomography in collaboration with Professor Pei-Jing Zhang from Oxford University. So we can get the first high-resolution 3D structure of the carboxysomes, including the rubisco packaging inside. So you can see there's two colors. The yellow one indicates the central rubiscos that are densely packed to form like a spiral structures, and the blue ones are the Rubisco enzymes that are close to the shell proteins. So they are relatively more random distributed in the carboxysomes. So with these structures, we can also study the interaction of the Rubiscos between neighboring Rubiscos and the key residues that are driving the Rubisco-Rubisco interactions. And interestingly, we can see Rubisco, they have a specific angle in the central regions. They have about 7.3 degree of twisting to determine the overall spiral-like structures. And we found two different organization of the carboxysome Rubisco organizations. So we further look into details and identified extra densities around Rubisco at the outer layer of the Rubisco regions. So this extra density hasn't been identified in the inner Rubisco regions. And based on the resolution, we can also identify these regions are a protein we call CSOS2, which is a linker protein, and also is a structurally disordered proteins that can recruit foreign enzymes into the carboxysomes. And the N-terminal domain is binding to the Rubiscos. And in vitro experiment also shows this linker region can trigger the formation of liquid phase, liquid-like condensates of Rubiscos, and indicating it's important for the cargo encapsulations in this protein organelle biogenesis. So we're also interested in the shell organization. Yeah, so using atomic force microscopy, you can basically look at the surface of the carboxyl shells and get a very low resolution structure of the hexamer and pentamer organizations. But because of the the softness of this carboxysomes, when we did the AFM imaging or nano indentation, we found it's different from the virus capsid, and it's much softer, 10 times fold softer than the virus capsid. So they have a specific assembly mechanisms, which make it challenging to see the overall shell structures. But we try to have a 2D layer of the hexamers to form a flat sheet, and using AFM, we can look at the different surfaces of the hexamer proteins. And we also conducted high-speed imaging to look at, for the first time, the assembly dynamics of the shell proteins to form the large facet. Okay, so to try to get a 3D structure of the shells, then we use nanobiology and building mini shells, which means we can select specific shell proteins like one pentamer and one hexamers. Then we can express them in E. coli to build mini type shell structures. So using this only two components, we can get about 22 nanometer or 25 nanometer shell structures, and which is much smaller than the native, about 100 nanometer. By adding this CSOS2 linker protein, you can get about 37 nanometer structures. Using cryo-electron microscope, we can get high resolution and study the variations of the shell protein interactions, for example, hexamer and pentamer interactions, and the hexamer interactions, and also the flexibility of the interface between the shell proteins, which indicates or determine the different curvature of the shell structures at different sizes. And very interestingly, when we integrate the CSOS2 into the shell construct, and we can see the direct interaction of CSOS2 with the shell protein inner surfaces. All right, so previously we mentioned the N-terminal is binding to the Rubisco, and now with the cryo-EM structures, we see the SC terminal of CSS2 is binding to the shell protein, and all the fragments, there are three CSOS2 C-terminal fragments that sit on the interfaces of neighboring shell proteins to combine all the different shell building blocks together to induce the shell assemblies. So based on the structure analysis, we now have much better understanding of the encapsulation and shell assembly mechanisms. For example, the CSS2 is a linker protein that using the N-terminal to bind the rubisco to introduce the rubisco into the carboxysome, and C-terminal is driving the assembly of shell proteins. If you look at the top view, and it's very similar to the footballs, that there are several building blocks need a specific thread to stitch all the shell proteins together. So the CSS2 is a molecule thread to bind all the shell proteins together. So in terms of the shell permeabilities, we use molecular dynamic simulations to look at a molecule, how much energy is required for for the molecules to go through the pore of the shell proteins. And basically, we found the negatively charged molecules requires less energy to cross the shell barrel, while as the oxygen and CO2, that's neutral molecules that needs more energy to cross across. So that also indicates the potential mechanisms of the selective permeabilities, but allows the accumulation of bicarbonate inside, while they can exclude oxygen, influx of oxygen and leakage of CO2 from the carboxysomes. We also developed the experimental techniques by building an empty shell and recruit a pH-sensitive fluorescent reporter to indicate the internal pH and external compared to the external pH environment. We found the shell organization can always create a low pH environment, and also up to 15 millimolar of bicarbonate can be accumulated inside the carboxyl shells, which is very important for the cargo activities. So now we have this better understanding or fundamental knowledge of the carboxysome assemblies. We are more confident to engineer this bacterial carboxysomes into the high plant systems. And also this carboxysome-based CCM system also provide a very promising target for enhancing crop plant photosynthesis, because the bacterial rubisco has a faster carboxylation rate than the plant rubisco. And also but on the other hand, they have a lower specificity to CO2 than oxygen, but this can be complemented by enhanced by the shell encapsulation. So the rubisco can be encapsulated in the environment that has a high CO2 to improve the carboxylation. So to test the hypothesis, we first engineered the free bacterial rubisco into tobaccos. Using the ballistic chloroplast transformations, we transfer a plasmid that express the bacterial rubisco large subunits and small subunits to replace the endogenous tobacco rubisco large subunit genes. And so we can get our homoplasmic transgenic lines. And the transgenic plants will grow autotrophically in the soil, in air supplied with 1% CO2. And the produced bacterial rubisco are mostly in the soluble fraction, and using cryo-EM, we can identify they have a typical decimer structures, and they are not affecting too much about the chloroplast structure, and because they are mostly soluble and never form aggregations. And we can detect activity of the bacterial rubiscos. You can see basically they have a twofold faster carboxylation rate than the native tobacco rubiscos. So we compared the growth of wild type and genetic plants in air and CO2 condition. So the transgenic plant shows essentially the same growth rate as the wild type in air supplied with 1% CO2, but could not survive in ambient air conditions. And the gas exchange experiment revealed that the net photosynthetic rate of transgenic lines was lower than the wild type. Nevertheless, our results demonstrated the catalytic activity of the bacterial rubisco produced in the tobacco chloroplast can support the autotrophic growth of plant in 1% CO2. This also highlights the necessity of engineering carboxysome in chloroplast. So previous work has shown the stepwise process of engineered carboxyl components, for example, rubisco plus the linker proteins, or rubisco linker protein with one shell proteins. They can form aggregations or some carboxyl-like structures, but it's not fully functional, and we need an intact and functional carboxysome to be expressed in the chloroplast. So to generate functional carboxysome, we generate a construct that includes 9 different carboxyl protein, the genes encoding the 9 carboxyl proteins, and transform into tobacco chloroplast. Hence, electron microscopy on the leaf of chloroplast shows there are indeed the carboxyl-like structures formed, and we have identified the further isolated carboxysomes and did the biochemical analysis using SDS page from immunoblot and mass spec to confirm all the 9 carboxyl protein can be expressed and integrated into the isolated carboxysome. And we also proved the functionality of the expressed bacterial carboxysomes. So the transgenic plant demonstrates the continuous autotrophic growth and a full life cycle, and in air supplemented with 1% CO2, which is supported by the produced carboxysome, but still not in ambient air conditions. And you can see the net CO2 assimilation rate of the transgenic plant was still very low. And this may suggest that the bicarbonate concentration within the chloroplast could be an essential factor for efficient CO2 fixation in the chloroplast producing plant. And the endogenous chloroplastic carbonic hydrate might need to be removed to keep the CO2 in or bicarbonate inside the chloroplast. Nevertheless, our studies show for engineering functional carboxysome in the chloroplast is an important first step to engineer a functional CCM system into chloroplast as a novel strategy for enhancing crop photosynthesis. So to build a functional active CCM systems, we also need bicarbonate transporters, for example, to be installed into the chloroplast envelope. And for that, we are collaborating with Professor Peng Zhang in Academy of Science, Chinese Academy of Science in Shanghai, to study the structures and regulatory mechanisms of bicarbonate transporters in cyanobacteria. We also need to plan to remove the carbonic hydrates from the chloroplast. So eventually we will have a chloroplast that can express carboxysomes and also transporters, but without endogenous carbonic hydrates. So the carbonic hydrates are fully encapsulated into the carboxysome to provide CO2 around the rubisco. So you can imagine that this is a long-term project that needs various expertise and international collaborations. And luckily and importantly, we have made an exciting start and are on the way towards a research direction that we think may have a notable scientific and economic impact. With that, I would like to thank all my group members now who are working very hard on this challenging project, and also the collaborators from many different countries and also funding agencies to support our research project. And thank you very much for your attention. I would like to take any questions.
Okay, thank you. Thank you, Dr. Liu, for sharing the very interesting work. Now let's start with the section of Q&A. So is there any question?
Okay, Lu Ning, very interesting talk. So actually, I'm very interested in the last part of your talk. So you engineer the functional carboxysome into the chloroplast. A simple question: which generation of the transgenic plants you used for your experiments? So another question is, so the chloroplast transgenic plants can be stably inherited.
Okay, yeah. So the first question is, there are two different, transgenic plants. So first one is just expressing the Rubisco. We got the hormone plasmid in the second generation. So in the next generations, they are all homozygous and they can be constantly used. And for the carboxysome, it takes longer time. So probably we tried have 4 generation or 5 generation. After that, it will be stable.
Okay. So
yeah.
After several generations, it's still very stable.
Yes, very stable. So we can always using the seed to grow. And we can do further, transformation to adding extra components inside.
Okay, thank you. So related to this question, the engineer make the crop plants to transform the chloroplast very difficult. So how do you think? In the future, how you engineer chloroplast for enhanced photosynthesis?
Sure. Yeah, this is a very good question. We are thinking about it. At the moment, we are still, it's a kind of a proof concept that we can engineer in tobacco as a because the chloroplast transformation is well established in tobacco.
Yeah.
And we are also thinking about other, crop species that can be, that is possible for chloroplast engineer. But along with these directions, we are also trying to develop the nuclear transformations to express all the necessary proteins in the nuclear using nuclear transformation and using, peptide to direct these proteins to be assembled in chloroplast. So that's another way, probably can be more extendable to many crop species.
Okay, thank you. Thank you very much.
Thank you.
Yeah, maybe I have a question, because I'm totally not into this field, but I'm just wondering, so the whole system with this carboxysome works when you have this higher level of CO2, left 1%, and doesn't seem to work with the ambient air. So means that you need to create a kind of accumulation of CO2 in the chloroplasts, and you are looking into transporters in the chloroplast membrane to engineer them or whatever, or increase the abundancy of these transports so they get more. But just wondering, will there be not more needed than that? Because in plants you have in the leaves you have they have anatomical histological factors that contribute to CO2 accumulation, like this Kranz anatomy and that kind of stuff. That and I'm wondering if it's possible actually to increase levels within the chloroplast without taking into account the CO2 concentrations in the leaves and the intercellular spaces. So maybe you need more than just doing that. I don't know. It's just
okay. Yeah. So that's really a good question. We also have thinking about this for a long time. Of course, there's no clear answer at the moment. But based on previous study using computational modeling, they think people think this transporter will have major effect on this bicarbonate concentrations. But what we try now is try to first to remove the carbonic hydrates instead of installing the transporter. So we hope in this step we can have a major effect on the local concentration of the bicarbonate, and probably the bicarbonate transporter might help further, but it's also technically more challenging. But people, our collaborator Maureen Hansen had already knocked out the carbonic anhydrase. So we are trying to collaborate to put our carboxyl in this species to see if that works.
Okay, thank you. Thank you very much.
Yeah, thank you, Tom.
Okay, if there's no questions, thank you, Dr. Liu again.
Yeah,
Okay.
thank you very much.